Hello, my name is Sebastian Aladin,
My name is Julia Higgins.
My name is Stephanie Espinosa.
My name is Carlos Hernandez.
and we're going to be talking about COVID-19 is virology, is epidemiology, and prevention. So let's give a little background on COVID-19. So COVID-19 is caused by the SARS-CoV-2 virus, and that is a coronavirus that was first identified in Wuhan, China in 2019. Because this virus was very infectious, it caused major outbreaks all over China, and then that caused that pandemic all over the world. In March 2020, the World Health Organization announced that COVID-19 as a global pandemic. The pandemic had more than 770 million confirmed cases and approximately 6.9 million deaths around the world, showing that the impact of this virus had globally. So this is the virus basics of SARS-CoV-2 virus. It is an RNA virus in the beta coronavirus genus. In several non-severe cases, it causes mild respiratory symptoms like coughing and a sore throat, while also causing severe headaches and diarrhea and body aches and exhaustion. In the more severe cases, though, it causes a loss of smell and taste and even pneumonia. So in this presentation, we'll be going over its virology, which is the structure, the genome, and the variants of the virus. We're going to go over its epidemiology, which is how it's transmitted, its dynamics, and its global trends. And we'll be also going over its prevention, which is the breakthrough of the mRNA vaccines and other things to do to keep yourself and your families and your friends safe. So the virology of SARS-CoV-2. So there are four structures that's important for this virus. All right. So we have the spike glycoproteins, which in this figure you can see them. They're the turquoise and orange parts. It's lipid envelope, which is the blue sphere, as you see in this figure that I got off Wikipedia, and is nucleocapsid, which is like inside his membrane, which encloses 30 kb single-stranded RNA. Oopsies, sorry. As well as non-16 structural proteins that are necessary for replication. So this is the the COVID-19 binds and enters your cells. It enters the cells using a spike proteins, which binds to your cells ACE2 receptors. And also uses your cell's protease enzyme TMPRSS2, and that like cleaves this RNA inside your cells. This is the most notable this entry is most notable in cells in the respiratory tract since they express the most ACE2 receptors. The strong affinity for SARS-CoV-2 virus for its ACE2 receptors kind of explains its high infectivity and its causes for respiratory illness. Because again, those cells in your respiratory tract have those receptors. Vaccines and antibodies that are effective because they target the spike protein receptor binding domain and fusion machinery. So now the genome and replication. This virus is a positive sense RNA virus. When it enters the host cell, its genome is released into the cytoplasm and then immediately translated into polyproteins. The polyproteins are then cleaved by, like I said, that protease enzyme into 16 non-structural proteins. Which make up the replication transcription complex. Synthesis of the full viral RNA happens in the host double membrane vesicles. This is a very efficient replication cycle, so this allows it to have very high loads of viruses inside the host infected cell. So now the mutations. Over time, COVID has gone through several mutations. Notably, the mutations are in its spike proteins, which like enhances infectivity. The variant, there are like a few variants of the viruses, notably alpha, beta, gamma, delta, and omicron, and they all have their own spike protein mutations. Alpha, beta, and gamma have the N501Y mutation, which improves their ACE2 binding to the receptor, and the P681HR mutation in alpha and delta variants improve their cell entry. These mutations drastically increase the virus infection rate and its spread. So now here's the evolution of COVID-19 throughout the pandemic. Because of the many variants that I mentioned in the last slide, but more notably Delta and Omicron, Delta was first identified in 2020, and because of the mutations, Delta became a dominant strain in 2021, causing massive hospitalizations and increased number of cases. Omicron, which was discovered in late 2021, had around 30 mutations in the spike proteins. This caused it to bypass your immune system like nothing we've ever seen before. Omicron evolved into many subvariations, and because of that, it became to dominate the globe. Like that was the main variant of viruses in 2021. Studies have shown that Omicron replicates in the upper airways though, and not in the lungs. And this is why it was less severe during the later parts of the pandemic, but it was very infectious. And now we'll be talking about epidemiology.
Okay, so when referring to the epidemiology of a virus, transmission is a crucial aspect to understand. Knowing how the virus is most likely to spread allows for better control of the virus and its lifespan. The dominant route of the spread of COVID is aerosol transmission. This form of transmission involves the virus being carried on small particles released by the respiratory tract. Both close proximity and prolonged exposure duration are key for transmission, which is consistent with most respiratory infections. While asymptomatic spread does occur and was an important factor to consider during the height of the COVID pandemic, studies determine it to be a less effective mode of transmission, being approximately 60% less infectious than symptomatic cases. Variable factors to consider in regards to transmission relate to patient age. Older subjects have been more likely to suffer severe consequences and are more likely to transmit the virus regardless of symptom status. Age and other risk factors we're going to discuss a little bit more in depth in the later slides. Surface or fomite transmission is also possible, but accounts for very little infections. According to studies, surface transmission rates are actually very difficult to quantify, as other possible causes of transmission must be considered possibilities, such as asymptomatic transmission. This being said, according to a CDC study, case reports suggest that surface transmission are less than 1 in 10,000. This scenario requires an infected person to cough or sneeze on a surface, releasing the aforementioned respiratory droplets, and another person must come into contact with these droplets that are on the surface and then directly touch their mouth, their nose, their eyes. Furthermore, these instances are less possible on outdoor surfaces due to air movement and sunlight. Okay, so one of the most effective ways that we are able to assess the transmissibility of a virus is by using the basic reproduction number, symbolically represented by R0 or R0, but it's pronounced R0. To explain this number with context, when the value equals to 1, this means that one person on average will be infected per infectious case. This value specifically is most easily applied in the face of a novel virus like COVID was, when a population has not yet been vaccinated and spread is uncontrolled. When other factors come into play, they must be taken into consideration during the calculation of values such as R0. The basic reproduction number varies slightly depending on the time of the studies conducted and their rollouts. However, it seems that the virulence factors worsened when COVID mutated and adapted into new variations. As you can see, Omicron stood out as a significantly more contagious strain with an R naught of 9.5 to 10, meaning that 9 to 5 people were infected from one original host on average. To understand the values used to describe viruses and their statistical profiles, close inspection into the mutations and variants that occur are very important. Medical intervention, like various treatment methods and vaccinations, affect how these numbers change and can also give rise to changes such as the Omicron and Delta strains. The effective reproduction number, symbolically represented by RT, essentially achieves this very notion. It is a way to assess the transmissibility of a virus while taking into account a population where individuals are less or simply no longer susceptible to the virus in the face of vaccines and previous infection, so antibody accumulation. Go to the next slide. So we're now going to shed light on some possible risk factors associated with the virus. As mentioned previously, age is a significant risk factor and plays one of the largest parts in the transmissibility as well as the outcome of COVID. Studies have shown that younger children and infants have lower transmission rates, meaning they are less virulent hosts than older adults. They also suffer less severe symptoms and oftentimes make speedier recoveries than many other age groups. Adults 65 years and older have been noted to have the highest rate of transmission, as well as representing the highest hospitalization numbers and mortality rates. These risks increase concurrently with increasing ages. Pre-existing conditions are incredibly important also to consider when analyzing risk factors associated with COVID. The CDC lists an exhaustive number of conditions that, when in combination with infection, can worsen the virus's symptoms. While there is a long list of diseases associated with the increased risk factor, the most important ones tend to be obesity, cardiovascular disease, both type 1 and type 2 diabetes, hematologic malignancies or blood cancers, and chronic respiratory and lung diseases, including but not limited to COPD, which stands for chronic obstructive pulmonary disease, pulmonary hypertension, and just normal pulmonary disease. COVID has also spread awareness on another risk factor, that being race and ethnicity. While certain racial and ethnic minority groups have been disproportionately affected by the virus, these are not necessarily correlated with genomic or biological differences, but instead socioeconomic barriers and systemic inequities. Lack of insurance and readily access to healthcare, crowded living environments, in addition to the inability to take time off work, puts these groups at higher risk of progressive disease and hospitalization. Although this topic may not be directly scientifically relevant to the virus's characteristics, it underscores a crucial aspect of epidemiology and how societal structures impact the life of the virus. The image regarding age groups and comorbid conditions is a visual representation of the aforementioned risk factors. As you can see, older age groups are represented much more in higher caseloads, according to the CDC.
In this slide, I'm going to discuss the preventive measures that have worked during the COVID-19 pandemic. It is more like a multiple defensive ways working together to attack the infection rather than just a single one. Among the 5 major ways I will talk about are social distancing, washing hands, wearing masks, immunizations, cleaning, and ventilating. The slide's graph shows the COVID-19 vaccine success rate of the 3 vaccines, Pfizer, BioNTech, Moderna, and Janssen, for both and partial vaccine types for the age groups of 65 to 74 and over 75. The whole of all vaccines is the administration point that is also the control on impact for both age groups. The Pfizer, BioNTech, and Moderna vaccines are over 90% impactful. The elder people over 75 years of age have a little more trust variety, where to say that the Janssen vaccine is still very good, notwithstanding its slope. Also, the utility is even low due to the primary partial immunization, while the range of proficiency in the group over 75 is relatively more, and it is less consistent certainly. Mainly with the vaccinated of both doses, the mRNA vaccines like Pfizer and Moderna play a bigger effect for the elderly regarding protection from COVID-19. The immunizations have the most efficacy way for the COVID-19 pandemic. The New England Journal of Medicine, a reference in PubMed, showed findings of how the Pfizer BioNTech mRNA vaccines with a 95 successful rate against symptomatic COVID-19. mRNA vaccines play a role in training our immune systems with the spike protein. They first figure out the protein spike, but they don't only do that, they teach our cells to create it so one's body is familiar with identifying and eliminating the actual infection whenever one has it. A new type of booster, due to the virus mutating, has become more mandated, which is the Omicron, mainly in the individuals with more likely risk, like the elderly and immunocompromised systems, where the doses are very effective and play a key role in their antibody levels. SARS-CoV-2 is distributed by the air, and this can be avoided from masks, mainly the masks that are the filtration ones and the KN95 ones. An article study done in 2021 had findings that masks decrease the incidence of COVID-19 by 53%. These findings of this research was made in different countries and environments that showed that just by covering your face is one of the most simplest but effective ways of stopping it. In addition, maintaining consistency is important. Research studies have shown that the impact of wearing masks in preventing COVID-19 transmission improves largely when a lot of people do it and is used correctly by covering the mouth and nose. Although SARS-CoV-2 is mainly transmitted through the air, the ways of hand hygiene have played a big role in places where a lot of people are in contact. As reported in the review article, soap and hand sanitizers break down the lipid layer of the virus. Therefore, these disinfectants can be an effective measure for preventing the virus from continuing its life cycle. There were campaigns of public health after the pandemic that established a rule of hand washing for 20 seconds. Hand sanitizers were made in public areas. These simple but effective ways of preventing it played a big role of the decrease of the infections. Social distancing, which was a rule made to be at least 6 feet from one another, also brought down and reduced the transmission of air droplets between one another in compacted areas. A worldwide research conducted in 149 countries proved that the social distancing measures indeed are linked to the large amount of declines in COVID-19 incidence rates. A case in point, nations that decided to close schools and workplaces firstly witnessed a rapid drop in the daily number of new cases. Long-term situations were more beneficial because of the social distancing that not only provided us time for testing, hospital capacity, and vaccination supplies, but also assisted us with them. In the end, ventilation and surface disinfection are two major environmental strategies. Indoor air that is inadequately ventilated can be a breeding ground for the SARS-CoV-2 virus. A PubMed article claims that airborne transmission is more common indoors and that using HEPA filters and opening windows to increase airflow notably lowers the airborne virus concentration. Additionally, even if the transmission of fomites is less manageable, routine cleaning of frequently touched surfaces, especially in the public and healthcare settings, helps to add an extra layer of protection.
Okay.
To begin the discussion on case studies and the rapid treatment strategies that were evolving at the height of the pandemic, we're going to examine this case study from April of 2020 about novel treatment strategies in an immunocompromised patient. The patient in this case is a 53-year-old male who developed textbook COVID-19 symptoms after international travel to Austria. The patient had asthma and was using inhaled steroids as a treatment, as well as oral prednisone for a prior upper respiratory tract infection. These prior issues suggest that he is immunocompromised, which potentially contributed to his worsening progression with the virus. The patient was experiencing fevers, cough, shortness of breath, and low oxygen levels. His initial PCR was negative, but the second test was positive, implying that the virus has variable detectability and that samples in the lower respiratory tract would yield more accurate results due to a higher viral load. As an initial treatment, the patient was placed on oxygen and given paracetamol, chloroquine, azithromycin, and lopinavir or ritonavir. All of these medications were initially recommended for treating COVID-19 infections at the beginning of the pandemic. The prednisone that the patient was taking for his upper respiratory infection, however, was suspended, which is also standard COVID protocol at the time. Corticosteroids like prednisone were actually recommended against at the beginning of the pandemic because in previous coronavirus outbreaks like the SARS outbreak, the use of steroids actually worsened outcomes and was thought to suppress the immune system too much. Despite following initial guidelines, he developed acute respiratory distress syndrome or ARDS and signs of a cytokine storm. The cytokine storm is due to an overproduction of pro-inflammatory cytokines. IL-6 was the focus in this case, and is part of the second or third phase of the viral infection. Cytokine storms can lead to severe inflammation, tissue damage, and organ failure, as shown by his severe inflammation and lung damage. This shows the life-threatening pathogenesis of COVID-19 virus, especially combined with previous risk factors like the patient's asthma. The patient was worsening despite standard protocol, so they revised the treatment to a more individualized approach. In response to his worsening lung condition and cytokine storm, he was given tocilizumab, which is an IL-6 blocker, and methylprednisolone, which is a steroid. His initial medications were stopped due to his heart rhythm risk, so the antivirals that he was on at the beginning of his treatment were stopped. This treatment was novel at the time because corticosteroids were recommended against with initial COVID treatment guidelines, as mentioned earlier. After the tocilizumab, his fever lessened and his oxygen saturation levels went from an initial 86% with room air to 90% with room air. The patient showed rapid improvement after treatment, and oxygen levels rose to 92% on room air 7 days after tocilizumab. His outcome was a positive one, avoiding mechanical ventilation and generally making a complete recovery, suggesting that timely intervention, targeted immunomodulation, and individualized treatments are effective in an immunocompromised individual. The photo on the right is a portable chest X-ray of the patient, showing progressive bilateral pulmonary infiltrates, which is significant and progressing lung inflammation or inflammation in both lungs. Next slide. Thank you. The next case study we would like to discuss is about an mRNA Moderna vaccine trial, and it was published in February of 2021, just shy of 2 years after the initial COVID-19 outbreak. The goal of this study on the Moderna mRNA vaccine was to prevent COVID-19 illness with onset in patients that were not previously infected with the virus, at least 14 days after the second dose of the mRNA vaccine was given. At the time of the study, public health protocols for preventing COVID-19 relied mostly on social distancing and masking, and this study shifted the global focus to mass immunization as the primary prevention strategy against COVID. The study also provided one of the largest and the most diverse data sets at the time, setting a benchmark for ongoing trials about the vaccines. It had a randomized model with 30,420 adults across 99 sites in different states across the US receiving either the vaccine or a placebo vaccine. The volunteers were randomly assigned a vaccine, and they followed through with 2 doses, 28 days apart. The study was observer blind, and it was stratified for racial and ethnic minorities and people more at risk in their communities. The goal was to fully understand how to provide the vaccine to the public equitably, focusing on the communities that might need it the most. The researchers did frequent follow-ups, adverse event reporting, and RT-PCR testing, all of which would determine eligibility to continue in the trial, ensuring the most accurate results. The image on the right shows how many were included and excluded from the per-protocol analysis and the individual reasons as to why. Persons would be excluded from the analysis if they acquired the virus before the trial was complete, missed the second dose vaccination window whether they were receiving the real vaccine or otherwise, received an incorrect injection, or they opted out. About 14,000 participants on the placebo and the vaccine side were completely vaccinated and included in the per-protocol analysis. As for understanding the molecular mechanism of the virus better, This study confirmed that targeting the spike S protein and its receptor binding domain via lipid nanoparticles is effective in treating the virus because it enables the delivery of the material needed to produce an immune response, directly blocking the virus's entry into a cell. The way the vaccine does this is by using a lipid nanoparticle to deliver synthetic mRNA, which encodes the perfusion-stabilized spike protein. This then generates neutralizing antibodies and a Th1-based immune response. This type of immune response is favored over the Th2 skewed immune responses that have been linked to vaccine-associated enhanced respiratory disease. No cases of this were observed with the Moderna mRNA vaccine from this study, which eased a lot of worry about prior vaccinations that were created for previous SARS outbreaks. The results of this study also suggest that early immune priming can actually reduce viral load and lessen severe outcomes in patients. As for the efficacy and safety, the mRNA vaccine demonstrated a 94.1% efficacy in preventing symptomatic COVID-19 and a 100% efficacy against severe COVID-19. All 30 participants who developed severe COVID-19 were indeed in the placebo group, with one passing away. Out of the 14,073 participants in the placebo group, 185 contracted symptomatic COVID-19, in contrast to only 11 participants in the vaccine group. There was a high efficacy rate observed across different ethnicities, risk factors, and previously infected people. There was no evidence of respiratory disease or major safety issues due to the vaccine, besides mild local and systemic reactions such as mild injection site pain, fatigue, myalgia, and headaches. It is important to note, however, that there have since been investigations and follow-up studies concerning the vaccine's long-term side effects, boosters, and dosing. This vaccine by Moderna was the first mRNA vaccine, along with Pfizer, to demonstrate such high efficacy in humans. Careless with how fast the vaccine became available to the general public, which was around 11 months after the virus genome sequencing, this vaccine revolutionized the speed and scalability of mRNA vaccine technology. The mRNA vaccine changed the course of the pandemic with mass immunization and shifted public health guidelines from containment to proactive prevention.
We all hope you enjoyed our project, and this is the final slide we have made with a summary of all the areas we discussed, which were virology, epidemiology, prevention, and case studies. According to the virology, the COVID infection SARS-CoV-2 is made from an RNA virus that is a single-stranded positive sense. It has different types of proteins, and it is a member of the beta coronavirus type. This pathogen can connect to the receptors of ACE2 on our cells and find its way through to effectively multiply due to the spike protein. It is a very contagious virus that goes through respiratory pathways. We then went over through its mutations and forms. There was a lot of mutations that was emerged as COVID-19 increased. There were ones like alpha, theta, and omicron types that would attach to host cells due to their mutations in their spike protein. P681H/R, which let the cells enter more accessible, where then N501Y made better ACE2 binding. Delta and Omicron, because of these changes and other variations, were distributed easier, causing more severity into the illness. It was also talked about the replication of the virus and genome. The viral RNA is sent out once inside the host cells and immediately then altered to polyproteins. The virus can make a large amount of RNA protein because of the distortion of the proteins that make the replication transcription complex. Symptoms appeared rapid due to the efficient replication and the high viral loads that were brought by the individuals who were infected and the ability of the virus to distribute quickly. Then came epidemiology, which we went over the global spread of the virus. In the beginning, it was thought that the reproduction number, or RO, which is how many people an affected person can infect, would range from 1.4 to 1.5, but it was increased, reaching 8 for Delta and 10 in Omicron, when the virus was emerged and developed into new variants. This quick increase shows the effect of viral changes and trends of our population. Regarding the air droplets and aerosols being the main way of infection, asymptomatic transmission was a big factor in the start of the spread of the virus. We then saw how surface transmission is a big key in infection. Unlike air transmission, although the virus can be on surfaces for a certain amount of time, We then looked at the factors of risk and its demographics. It was shown that the elderly and those with critical conditions and immunocompromised are more to go through it more serious and life-threatening. However, on younger individuals, it was tended that they have not as severe symptoms, regardless of them being able to give the virus to others. We went over several preventive ways of this virus. The most effective one was being immunized. It was shown in the findings on 86% to 93% success rate against symptomatic and close to 100% of effectiveness towards severity cases. Vaccines like Pfizer and Moderna were impactful for the rapid evolution and levels of success. Wearing masks, social distancing, washing hands, and correct way of cleaning and ventilation were also all important in ceasing the spread of the disease besides immunizations. Finally, we also looked into the case studies where in this case, the first case, there was a patient immunocompromised that gave a good response that was targeted to treatments such as IL-6 blockers, emphasizing the effectiveness of customized therapy that was tailored to them. A second case study, which was a mRNA more than a vaccine study, was a big study with a large number of individuals across US sites. It proved that with the mRNA delivering coding for the spike protein through lipid nanoparticles, successfully produced a strong immune system. The immunization showed a 94.2 successful against symptomatic and 100% with critical cases. This played a big role in improving and rapidly increasing the development of vaccines and prevention through large immunization. Moderna mRNA vaccine, a novel approach to effectively preventing illness and modifying the path of a pandemic, was the second case study. The prevention and treatment strategies, which were based on our knowledge of the virology and evolutionary biology of SARS-CoV-2, saved millions of lives. The global response to COVID-19 demonstrated the power of science, medicine, and collaboration in crisis management, as well as the importance of adaptability in the face of a constantly evolving virus. Thank you for watching.
All right. And these are our references. Hope you enjoyed. All right, perfect. Hold on.